Okay, great. So we are going to have a couple of folks do a brief presentation. This was at a request from our last meeting, where as we kind of work into the season, there's a lot of things that could be impacting different birds or marine mammals in the marine ecosystem this summer. And so it was suggested that it would be great if we had some folks who are experts provide some information about, okay, what are the symptoms that you might see in seabirds or marine mammals if they are being impacted by harmful algal bloom related toxins. And so we've had Kathi Lefebvre, who's going to chat with us about marine mammals, and then Sarah Schoen, who's also going to talk to us about seabirds. And so we'll have Kathi do a brief chat and then we'll pass it over to Sarah. Folks, feel free at once in between, we can help with questions. And then we'll kind of roll into round robin updates from folks as we have time after that. Cool. Looks lovely.
Okay, can you see that?
Yes.
Okay, great. Um,
Oh,
okay. Oh,
it's not in presenter mode, but we can see it.
is it big enough?
Yeah.
Okay,
Yeah.
great. We'll just do this.
Cool.
Um, okay, great. So I'm happy to be here. I'm going to share with you, I'm going to do a brief overview. Just tell me if I'm giving too much detail, but I wanted to share exactly what we're dealing with when we're talking about these toxins. So everybody already knows on this call, everybody already knows that the species of concern are the Pseudonychia and the Alexandrium. The Pseudonychia produce the domoic acid, and the Alexandrium produce a suite of toxins, with saxitoxin being the main one we think about. And they have two very different mechanisms of toxicity and therefore have very different clinical signs in affected animals and humans. And so don't be scared of this next slide. It's really cool. You're gonna find this interesting. I'm gonna start with domoic acid. Maybe. Okay. Let's see. Okay. So just to give you an idea of what's happening, up in the left-hand corner, these are two nerves. These are two neurons, cells. The first one signals the second one through an action potential. Okay. So domoic acid, just to give you an idea, see that little black square? Can you see my pointer? Okay, great. This is called the synaptic cleft. And when there's a signal or stimulus in an animal or a human, any mammal, or a bird, the signal goes down here, and then there's a release of glutamate. That's the normal thing. So what happens with domoic acid, if you go down this arrow, this is the synaptic cleft right here, which is where this little box is. Okay. This is the nerve that's sending the signal. What it does is it releases these vesicles that contain glutamate. The glutamate goes into the synaptic cleft and binds to receptors in the other nerve and causes an action potential. And that's just the normal process. That's how you do something. That's how you get a stimulus, and then you can make a movement. Domoic acid, these green things here, can get into the synaptic cleft and bind to the receptors, but binds longer and harder than glutamate. So it overstimulates the nerve. So that is the problem. Okay. So what I want to show you here, what does it look like in a marine mammal? This is a California sea lion. This video is taken by Dan Ayers, Washington State Department of Fish and Wildlife in 2015. This is a sea lion we know was poisoned by domoic acid. We got samples from this animal, very high concentrations of domoic acid. And this is something that's well known and very obvious, these kinds of symptoms. We get this. The first event that we heard about or that we documented was in 1998 in Monterey Bay. Since that time, there have been events, animals impacted every year in Southern California, Central California. Just to mention the severity of it, in last year, 2025, approximately 2,000 marine mammals were impacted by domoic acid poisoning and blooms. 350 of them were dolphins. So there's dolphins, seals, etc., sea lions. So it's a really significant thing happening in Southern California on a regular basis. Okay, so that's a domoic acid story. That's an impact in marine mammals, and it's a very significant impact to marine mammal populations in California. So let's move to saxitoxin, just to give you the difference in the mechanisms. So saxitoxin does the opposite thing. It blocks signaling. So I'm showing a little schematic here from a review. We've all heard that saxitoxin blocks voltage-gated sodium channels, which just means they can then not signal each other, and that causes paralysis. And so here's a sodium channel, calcium channel, and saxitoxin gets right in there and blocks them, so they cannot pass ions through and they cannot signal the next cell. What does that look like? So the domoic acid seizures, et cetera, are very visible. We've done some laboratory studies with a model organism called zebrafish. And this is what a touch response looks like in a normal, healthy zebrafish larvae. And this is what it looks like in a zebrafish larvae exposed to saxitoxin. So that just, you can see that there's not something so visible to see with saxitoxin poisoning in marine mammals necessarily. So just to reiterate, when you're looking at symptoms in marine mammals, they're going to be different for various toxins. And the saxitoxin, which is the most concern in the Alaska area right now, is less visible. And just a quick overview. You've probably all seen this before, but through our HAB program, we're seeing that if you look at these red symbols here, these are clams. Red means they have high concentrations of saxitoxin that would be considered dangerous. So we are seeing saxitoxin above seafood safety regulatory limits every year throughout northern and western Alaska. I don't have a detailed map of the Gulf because we've been focusing in this sort of new area. But we know that those exposures are there and beginning. We haven't, up until 2024, we hadn't had a confirmed marine mammal poisoning event from saxitoxin, and I'll talk about that in just a little bit. And I'm almost to the end here. This is also those same maps, those same symptoms with domoic acid concentrations. And you see what you won't notice right away, there's no red. So these concentrations of domoic acid in all years throughout all these same places are not near the seafood safety regulatory limits. So we're considering that as a lower risk right now. So here's just a general schematic for that. In the lower 48, domoic acid is the biggest concern for marine mammal health. It's a cytotoxin. We have events every year with thousands of animals coming up on beaches. It's very visible. In Alaska, the concern is more saxitoxin. There's more of that toxin there at toxic doses. And in 2024, in the Pribilof Islands, we had the first confirmed event in a marine mammal, the northern fur seals on St. Paul Island, that were poisoned by saxitoxin with a bloom. And there's a publication in Marine Mammal Science there that you can look at. Unfortunately, the next year, 2025, the same thing happened on St. George Island, very nearby. I'll tell you why that is really concerning, other than the obvious. So, last couple slides. Domoic acid poisoning in California sea lions was first documented in 1998. Since 1998, this has happened every year, and there's a statistically significant relationship in that it is increasing since 1998. I don't have the latest data points on there, but it is. Last year in 2025, when we said we had 2,000 animals in Central California, that started earlier than ever, February to May, and it was longest lasting that we've seen so far. And that was for domoic acid. So there is evidence, because we had a little scare last year in Kachimak Bay, thinking about pseudonychia, there is evidence for increasing domoic acid prevalence and exposure risks in Alaskan waters. So remember, this is the trend for domoic acid in Southern California, Central California. In the Bering Sea, we published a paper on ice seals in 2021 that showed a statistically significant increase in the probability of toxin being present in bearded seals over a 10-year period. I want to say, though, these are still well below concerning levels. These were very, very, very low levels. So I will go back to my summary slide here. So just where we are, the state of what's happening with marine mammals and algal toxins right now is that domoic acid, again, is more important or more significant impacts in the lower 48. Saxitoxin is more important in the Alaskan waters. It's obviously, we just had our first event. What's concerning to me is I was involved in the 1998 first documented poisoning event in sea lions with domoic acid in Monterey Bay, California. I was on the ground floor of that investigation. And then they happened every year after that. I've now been on the ground floor of the investigation of the first northern fur seal of a poisoning event on St. Paul Island, and now there's already been another one. So what does that mean? We'll be watching it, but it is, to me, showing that, yes, we need to be concerned about saxitoxin poisoning in marine mammals. We currently don't have those same kinds of concerns for domoic acid in marine mammals, even though we know that they're increasing in the background. And so therefore it's important to be monitoring and looking at it. And then Sarah's going to speak next about birds, and she had asked me to mention about effects in fish. So we do know that saxitoxin can kill fish. There's been some reports in the past by White in herring, massive die-offs due to saxitoxin poisoning. Domoic acid, was actually my thesis work in grad school, looking at why are fish not affected by domoic acid when marine mammals are. And that's really an interesting story. Anchovies, salmon, other fish that we've tested, when you feed them domoic acid, they can accumulate very high concentrations in their GI tracts, and they don't exhibit symptoms or death. So far, we have not seen die-offs due to domoic acid, which makes them, when they do get toxic enough, very dangerous vectors because they stay in the food web. So anyway, that's where I'll stop. And hopefully that also laid the groundwork for Sarah to talk about symptoms in birds.
Great. Thanks, Kathi. I think, Sarah, before we move over, I know there was at least one question in the chat, Kathi, for you from Rob Kaler with U.S. Fish and Wildlife Service. And it
Okay,
was curious what the level of exposure quantity for saxitoxins was for the zebrafish.
Okay. That zebrafish experiment was not injection. It was dissolved in the water. And I can send you the paper with those doses. We tried to target doses that would be realistic in the dissolved fraction in the ocean that could be that could occur with larvae floating in the plankton. So I don't know. I don't want to misspeak on what those concentrations are.
Kathi. And then also curious about how you determined the anchovy study.
Yes. So we So my first year of grad school, I was studying domoic acid, wondering why it doesn't impact anchovies, but we had a bird event. That's why I was already studying domoic acid when this newspaper article came out and said all these sea lions are having problems, they're having seizures, they think it's mercury poisoning. And I'm sitting there studying domoic acid and its mechanism. And I'm like, I think it's domoic acid. So I went to like the Marine Mammal Center where Francis Gullen was. I got samples. Anyway, with a bunch of really great people, we discovered that it was. So I did a lot of experiments with anchovies. I injected them with domoic acid. When you inject fish, anchovies with domoic acid, they have seizures. They do spiral spinning. They just freak out. They can't school. So I thought I had the answer. Yes, their nerves are susceptible to domoic acid poisoning. There it is. But we just weren't seeing it in the field. So then it's like, well, I have to do an oral exposure, right? They're not exposed by injection in the world. So I did. I drove to bait shops. I got fish in big circular tanks at Long Marine Lab. I cultured liters and liters and liters of pseudonychia and fed them and changed the water every 4 hours for 4 days, slept by the tank, trying to get a bloom. We did get some toxin, but it just wasn't a high enough to prove anything, and there were no impacts. Then I took 700 anchovies on a cruise on a research vessel, and I pumped on a bloom into a giant tank. And I will tell you that I carried those anchovies like 5 at a time in plastic bags to get them on the boat. It was a massive effort, and there was a bloom we got in there, but we saw no symptoms. And again, it was not that high. So then I moved to salmon, and I gavaged them with a gavage needle and to ecologically relevant doses, and did not see any symptoms. So there's a tremendous effort working with fish to try to determine if they have any impacts from the toxin. So most likely we're thinking that they have a way to sequester it to the bile and get it away from the central nervous system.
Yeah, that's awesome. Thank you, Kathy. And I think you've set Sarah up for her presentation. I see there's other questions coming in, but yeah, no, that's great. And if you, I guess, post your papers, that would be great. Very interesting.
Okay, should I send them to you, Rosie, to put on the? Okay,
Yeah, that would be great. Yeah, we can send them out in the notes to everybody if that sounds good.
sounds good.
Um, okay, Kathy, you have a couple more questions, and then Sarah will, I feel like once we do these questions, we'll pass over to you if that sounds good. So there's a question from Chris Holderreed, and it's saying, what do you know about domoic acid monitoring results in British Columbia and how far north the higher levels extend?
That's a good question. So our program here, the Warren West program that we've mentioned before, Wildlife Algal Toxins Research and Response Network, we get marine mammals. We include British Columbia, so we take animals from everywhere. And I'm contacted quite often from Canada or from BC for thinking about algal toxins. As I wish I was more up to date on the real details, but they're concerned about it. But on our West Coast, they haven't necessarily been having high concentrations. And Chris, I can get back to you with more details on that, but nothing like, on the east side with the Prince Edward Island event with the humans. So they're concerned about it, but they're not seeing stuff like we do in California with the
Yeah.
marine
Do
mammals.
you think the split is at the Columbia River and it's north isn't getting it and south is getting it?
I know, I've thought about that a lot. One thing I think is that it's there are 3 components. You need the bloom, very toxic. You need the planktivorous fish. You need the anchovies or the sardines or the herring to be accumulating. And then you need the marine mammals there, and they need to be feeding. So that all happens in California. That doesn't happen so much in Oregon and Washington and up north, as far as I'm seeing. And people who understand marine mammal populations and migrations and stuff could think about that probably in more detail. But I think it's a combination of all of those things. It's not the perfect storm farther north yet.
Kathy, I feel like we've had a lot of discussions about like where's the line, about like keeping an eye on it, as we're thinking about it up here in Alaska. Okay, great. There's a question also from Danielle Garrick, and it says, are there noticeable symptoms of saxton poisoning in whales?
That's a great, 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 great question. I'll tell you what I think. You saw that little zebrafish larvae not doing anything when we touched it with saxitoxin poisoning. What I think is that, same with the fur seals, northern fur seals, when an animal has a high enough dose of saxitoxin to cause paralysis, wherever it is, which is a whale is in the ocean, basically they're going to stop being able to breathe. So they're not even going to drown. They're not going to take in a breath, a blowhole of water. They're just going to suffocate. They're not going to be able to breathe. And that makes it less visible to us because they're out in really remote areas. And we do see, animals do come ashore. When you think about that St. Paul Island event, Benson Beach, 10 dead northern fur seals washed ashore. We did backtracking with Bob Picard on the oceanography of it to where they were feeding, showing that they could have suffocated where they were eating and been brought with the ocean currents the way they were working and landing on Benson Beach on the exact day they did. Mm-hmm. So if you think about that, we saw 10 animals that happened to hit this tiny little beach. How many animals do you think were impacted? I think that animals are impacted that we're not seeing. And that would also be the case for whales. However, with domoic acid, it's an excitotoxin. You get overexcited. You do see in dolphins, you see flipper movement and movement. For sea lions, they don't stop breathing. They come ashore and they start to do things. So when you ask for symptoms to confirm saxitoxin poisoning, it's going to be very difficult. Other than that.
Yeah.
Okay. And then one more question, Kathi, from another one from Chris is how long did the D levels stay high in the fish? And was it only in the gut without injection? I know that's been like a big conversation here is worried about like subsistence harvest, right? As you're used to marine mammal concerns, but with fish.
So in one of my papers from my very first year, looked at bloom densities in Monterey Bay and domoic acid levels in anchovies at the same time. And they were perfectly correlated. So the higher the cell densities, the higher the toxins. As soon as they went down, they were gone. So depurated very quickly in anchovies related to its exposure. When we did the gavage with salmon, we looked at all the different organs and did depuration over time and did blood draws. It was really an incredible experiment. And the blood draws were possible with Irv Schultz, who does that kind of stuff, repetitive blood draws over time. So it's depurated quickly, goes to the bile, etc. So that's the case there. Other things can hold on to, like razor clams can hold on to some of the toxin, more so with low levels of domoic acid, not high levels though. So yeah, fortunately they're water soluble and pretty quickly depurated for domoic acid. And the tissue fillets, so there might be 2,000 ppm in the visceral mass and 0.5 in the fillet. Just 1% of the total toxin.
That's awesome. It would be wonderful to have some of that kind of stuff summarized. Rosie, I'm kind of pointing for to have on the from your papers, because that detailed work is super hard. Those are amazing. I'm just amazed at the studies that were done then. To summarize, because this question comes up all the time, and having some of that, these are results that we're seeing from that time, summarized would be great. Thank you.
You know what I can do, Rosie, if you want, I can take some of those figures that are visual and make just like a bullet conclusion from all that for the various things for domoic acid. And then I'll send it, Chris, maybe I'll send it to you first to have you see what you think, you and Rosie. that could be good to put on the website with the references. And it's going to be a little while. I'm not going to do it tomorrow.
Kathi, that would be great. I think, like Chris kind of is already saying, is that whenever we have any concerns about that, one of the first things that comes up is people are worried about fish. And so I think, we have some like past studies where folks like Steve Kibler have done some food web stuff, looking and testing all the different organs and whatnot. We can point to kind of those studies, but I think having more resources for people that they can also look at things in a way that they can understand is always great.
And understanding the difference between saxitoxin and
Yeah.
acid.
Yeah, absolutely. Yeah. Okay, Kathi, thank you so much. I think we'll rotate over to Sarah to chat about birds now. Thanks, Kathi.
All right, can you guys see my screen?
Yes.
All right. So for those of you who don't know me, my name is Sarah Schoen, and I'm with the USGS Alaska Science Center in Anchorage. And I'm going to talk about behavioral effects of saxitoxin domoic acid exposure in seabirds. So Kathi already gave us a great overview of toxins, so I'll skip through the intro material, but just note that some bird mortalities have been attributed to saxitoxin over, the last many decades, but those are often correlative and lacking quantitative information, which really spurred this first dosing trial that Bob Jusek of the National Wildlife Health Center led on mallard ducks. And he published a good paper in Harmful Algae in 2021. So he found that at the acute dose of saxitoxin poisoning in mallards, death occurred quickly within under an hour. And for birds that survived that dosing, they didn't see clinical signs within 24 hours after dosing. So that toxicity effect wrapped up pretty quickly for birds that survived. But they did see a weight loss in birds that were dosed. Between 1.8 to 6.3% of body weight was lost. So they weren't eating as much after dosing. And some of the behaviors that they saw, head shaking was seen in all of the dosed birds, excessive drinking, regurgitation, although notably the birds that died did not regurgitate. So that we can infer that that's kind of a behavioral adaptation to limit toxicity. Wing twitching and settling was seen in some birds, and tail wagging. And then Matt Smith, formerly from the USGS Alaska Science Center, led a study on common myrrh dosing down at the Sea Life Center. And we published a paper in 2025 in Harmful Algae about that study. This had two different components. One was assessing the ecologically relevant behavioral changes, the dose necessary to induce those changes that would likely lead to death in the wild, as well as looking at chronic dosing effects on birds. And one of the main takeaways was that the behavioral response from saxitoxin could be difficult to observe. It was difficult to observe in these myrrhs in the lab, and it would be difficult to observe in the wild, like Kathi was mentioning, with whales or other organisms. And he found it was a really fine line between the subtle response to dosing and acute toxicity. At acute doses, death occurred quickly. This was even quicker than the mallard study, within 20 minutes. Abnormal behaviors began immediately post-dosing, and if birds survived dosing, those abnormal behaviors wrapped up within 30 minutes. So this was a really quick effect. And again, dosed birds in this chronic dosing study ate less per day than control birds and lost 5.5% of their body weight. And the one significant difference in behaviors between dosed and control birds were that dosed birds rested less than control birds. So some of the specific behaviors that were observed after dosing included uneasy posture, head shaking, regurgitation, wing abduction, and paralysis of the wings, legs, and head at extreme doses. And these are all Matt's photos from the study, and we gave a presentation together a few years ago, so I'm just using his animations here. But you can see that the circled bird, the circled myrrh here, has its wings out and, a very unusual posture, not grouping with the other birds in that photo. That's 34 minutes after injection. By 38 minutes, it's back in the water and kind of grouping together. And by 50 minutes, it's pretty indistinguishable from the other birds. And then that at 105 minutes, it's back to swimming in the water. And then it was observed catching a fish at 106 minutes after dosing. So we also have some information about saxitoxin exposure in wild birds and behavioral effects. Again, this is really hard to catch. You have to be there at the right time and place, and also collect samples that can confirm what was causing the effects. But there are a few studies that have seen regurgitation in wild birds associated with saxitoxin exposure, including from Kitlitz's Merlet chicks on Kodiak Island that were being fed pretty hot sand lance, Pacific sand lance. Weight loss has been seen in the wild, and that was again seen in the mallard dosing study and the murre dosing study. And then during the 2017 multi-species seabird die-off in the Bering and Chukchi Sea, there was a lot of different behaviors that were noted. It wasn't mentioned which species, but in that die-off, the northern fulmars ended up having pretty high concentrations of saxitoxin present and were later determined, some individuals were determined to have died from saxitoxin poisoning. And some of the behaviors noted in birds during this event across a really broad area of the Bering and Chukchi were weakness, lethargy, drooping heads, lack of predator avoidance, staggering, bobbing in the water, throwing themselves on their back and righting themselves, and swimming in an odd motion. Okay, and moving on to domoic acid. Again, Kathi set us up well on this, and I'm not going to go through the background, but again, similar to saxitoxin, bird mortalities that have been associated with exposure to DA are often correlative and lacking quantitative information until recently, and I'll talk about that later. There has been one dosing study that I'm aware of with birds and domoic acid, this PhD thesis in 2003, which used 3 different bird species, pigeons, mallards, and common murres. And overall, across these different birds, the birds showed signs of toxicity within 20 minutes of injection, and those clinical behaviors peaked around 220 minutes post-exposure. The surviving birds appeared normal within 6 to 7 hours. And the initial signs of clinical signs after this exposure consisted of apparent agitation, head bobbing, and increased unusual movements. And that was followed in more higher doses by extreme muscle tremors and abnormal stance, increased water uptake, and affected birds were passive and didn't resist being handled. So this, in the study, this figure here that just popped up shows some of those abnormal stances in the pigeons with their wings kind of held out in a weird posture. And then with the ducks, they would lay down, their heads were horizontal to the ground, kind of laying down, not moving. And then specifically, there were some of these behaviors that were specific to different species. The pigeons were doing head bobbing, pacing, circling, and seizures, muscle tremors in the wing and tail. They had a dropped hawk and wing posture, arched body position in extreme toxicity, increased water drinking, fluid discharge from the nose and mouth, and difficulty breathing. The mallards had some similarities, head bobbing, pacing, circling, more widespread muscle tremors, again, that horizontal posture and immobility, hyperthermia, failure to group with other birds, and decreased response to stimuli. And the murres were more similar to the mallards, with muscle tremors, that abducted wing with uncrossed tips, lethargic, riding low in the water, hypothermia, failure to group with other birds, and again, the decreased response to stimuli. And there have been domoic acid exposure in wild birds noted in the literature, but specifically increasing, like Kathi was mentioning with the marine mammals, in the last couple decades here. And those exposures with wild birds have mostly occurred in California and Mexico, including a recent outbreak in California last year. And the highest levels, DA levels, in seabirds were found in California in 2017. It's mostly affected cormorants, pelicans, loons, and grebes. And some of their behaviors in the wild have included seizures and tremors, disorientation and imbalance, difficulty walking or flying, and memory loss and behavioral changes, including unusual movements like flying inland. And this is a video, I hope it will play, there we go, by BirdLife International showing a seizure in some birds. And then lastly, I have not observed these behaviors myself, but I found this description by Corinne Gibble in her 2021 summary paper. She summarized some events from California that had birds had both saxitoxin and domoic acid present. And I'll just read the quote. Some of the clinical signs of domoic acid toxicity and saxitoxin toxicity in seabirds are similar, including vomiting, diarrhea, and death. But the hallmark clinical signs, seizures and tremors for DA and muscle paralysis for saxitoxin, differ. And with that, I'll take any questions if there are any.
Sounds good. Great. Thank you, Sarah. I know I had a immediate question that I was curious about, which was in regards to birds with domoic acid or saxitoxin exposure levels, will it work its way out of their system on their own if they're exposed at a higher level, or are you assuming that a lot of those birds will die?
Yeah, that's a tricky question. So these dosing studies, the mallard dosing study, it did show that at higher concentrations of saxitoxin that they were dosed, the birds died quicker. So there was a correlation in that regard, but there wasn't always a similar difference in concentration that you could find in the bird. So if you found a die-off bird and you looked at the concentrations within the tissues of the bird, they don't correlate, at least in the mallard study and the MERS study, they didn't correlate with the dose that they were given. So yes, I think the dosing studies have shown that at higher doses, birds will die quicker, but we can't back calculate that just by looking at a die-off specimen. So that's really tricky.
Yeah, Rob, go ahead.
Yeah, thanks, Sarah and Kathi as well. This is this is great. So Rob Kaler, Migratory Bird Management, U.S. Fish and Wildlife Service based in Anchorage. My question, and I'm not in the Dusick paper and the Smith paper might cover this, but I was thinking about the physiology of birds versus marine mammals, and that's why it's also nice to have Kathi on, because I think Sarah and I are both bird people. But thinking about like how birds have a crop, the stomach, you've got the proventricular and the gizzard, and apologies, I'm not an animal physiologist, and then the excreting of the waste, the cloaca, single exit. So how that might influence how these animals are coping with biotoxins.
Well, the regurgitation, I don't know about the physiology, the structure of the bird anatomy, how that affects it. But I do know that regurgitation is a behavioral adaptation. And that was something, I think that was part of your question, Rosie, that I didn't get to. But in all of these studies, they are finding that birds that are exposed to the toxin, especially DA, were pooping quickly. They were, excreting from both ends quickly. I think that's a response that they, for both toxins, the animals are trying to excrete this quickly. And it, like Kathi was mentioning, it's water soluble. And that is a good behavioral adaptation to a limit. But if they get dosed at too high of a dose, they could die acutely. And then again, with especially saxitoxin, when they are experiencing paralysis, in both of the dosing studies I talked about, they're not able to eat as much right afterwards, and that can quickly compound. If there's bad weather, it's cold out, birds have a really high metabolism. They have to eat so much, so quickly, like 60 MERS have to eat something like 60 forage fish a day to maintain their mass. And if they can't maintain their body mass within 4 or 5 days, they're going to die from starvation. So you could envision a scenario where exposure to a toxin could quickly cascade into a starvation event that you can't necessarily link to toxin exposure.
Yeah, Kathi.
Oh, I was just going to add that I remember working with Paul Silvagni, who did that study in 2003. And what was really surprising is that birds were more sensitive than mammals. They were significantly more sensitive to the toxin than mammals. And we have all kinds of ideas about like length of digestive tract and various things, trying to look at the comparisons between mice, monkeys, birds, marine mammals. But anyway, so they're very, very vulnerable.
That's really interesting. So presumably if they have a longer digestive tract, they're going to it takes longer to excrete the toxin. Is that the idea? And they might be more susceptible.
Well, I'm trying to remember the details of it, because I think that it may have, we've did so many studies, it may have to do with, where it's absorbed and et cetera.
Mm-hmm.
So I want to get that information correct, but I will also send a bullet on that for the summary.
Sounds good. Yeah, in that thesis I did read yesterday that the lethal dose for mice was much low, or much higher. So yeah, birds were more sensitive than mice.
And then there's a question in the chat. This is another one for Kathi. Does regurgitation also occur in marine mammals with saxitoxins?
That is an interesting question. So, when Sarah was mentioning how domoic acid and saxitoxin have similar, like diarrhea, vomiting, etc., those are just basic standard responses to poisonings, to getting, when you get something that you don't want in your body. So I assuming that they probably do. I don't have evidence of that. But we know that the sea lions, in the California with domoic acid have diarrhea, etc. So I'm sure that those normal symptoms from poisoning are occurring. We're just not necessarily seeing them. And then Chris, I'm noticing your question here on, yes. And Sarah brought that up with the memory loss and the birds going in weird places. And it made me remember that I forgot to mention that in the marine mammals, if they recover, they can have a chronic condition. There's been some absolutely incredible studies done, one by Peter Cook, where he looked at sea lions that had been naturally exposed, and he did MRIs at the hospital in live sea lions to measure their hippocampus. And then he did these training mazes at the with pools and having them make decisions to go to prey, et cetera, and found a direct correlation between the size of the hippocampus. The smaller, the more memory issues, the less ability. So there's permanent memory loss and behavioral impacts of that in marine mammals, just like we know in humans, there's permanent memory loss. And so we see things like during these events, we can see sea lions in artichoke fields in Castroville. We can see them on a cop car off Highway 1. We saw one animal go all the way through the San Joaquin River to Stockton. So, yes, they have permanent issues, and it affects their ability to obviously get food and to remember where they're going.
Yeah. And I know that we, at least from the samples we collected from Ketchikan Bay last year, the highest levels we saw in regards to domoic acid in shellfish, I think was from razor clams, and it was 0.2 parts per million. And when you start getting worried about these like long exposures to low levels, you don't get worried until you hit numbers like 2 to 5 was what I've heard from like Jerry Bosher at Department of Public Health. So I feel like when I was kind of thinking about this information about like, okay, could we be worried about marine mammals or birds or humans in our area and these like low level exposures to DA when it's that low? And he just let us know like you're not there yet, which is just some good information going into this summer about like, okay, what does low levels mean? There's levels of that as well. Okay, there's a question in the chat from Natalie to Sarah, and it's in Matt's study, did you test for saxitoxin in the GI content or feces by HPLC or ELISA after animals were dosed to see the difference in the dosage amount versus how much is later detected?
Yep. And that was similar to the mallard study. So it wasn't correlated directly. Matt didn't actually do an analysis on it because it was very, they weren't correlated. But yes, so the birds, there was a subset of birds that were euthanized after the experiment. And he looked at all of the tissue concentrations after dosing, as well as the birds that died acutely during the initial testing for the ED50, the ecologically relevant dose. And it wasn't, there were some higher levels in some tissues. The tissue concentrations weren't really predictable. He saw some tissue concentration, the highest toxin concentrations were found in the GI and feces, for sure. The feces had the highest, I think the highest was 450 micrograms per 100 grams. So much higher than we've seen in the wild. And the GI had high concentrations too. There were some concentrations found in the kidney and really trace amounts, and some in the liver, but lower than we've seen in the wild, which I'm assuming is related to, low subacutely lethal chronic exposure in the wild might build up in the liver and kidney. But yeah, it wasn't a direct correlation. So again, it's confusing when we're trying to back calculate the cause of death from a die-off bird on the beach.
Yeah, absolutely. And then before we wrap up, I have a question kind of for both of you, and this is a question I've been getting from folks, which is why I wanted to mention it, was there's obviously a lot of things that could be impacting marine mammals and seabirds, and especially this summer moving forward. And one of the ones that I've been hearing from people being worried is that is there a difference between symptoms for avian influenza versus potentially HAB-related toxins? And I was wondering if you both wanted to respond to this. I think we have some general messaging in regards to like, unless you are trained and have the proper PPE, you shouldn't be sampling marine mammals or seabirds that you find dead on the beach. But I'm curious in regards to potential symptoms, if there's a difference.
I can mention something. I was at a meeting several years ago, and someone sent a video of a bird in Alaska having issues, and it was tremoring and, doing these things. And I was like, oh, yeah, that looks exactly like domoic acid poisoning. And it actually was not. It was avian influenza. So yeah, that's a concern. I don't think you can be 100% sure just looking at this tremory behavior.
Yeah, I think that's a great synopsis. And I think we should get Andy Ramey to talk with us about some of the symptoms from HPAI. But from what I can remember him talking about recently, I think spinning kind of, I don't know if it's their whole body spinning or their head spinning. That was one of the really hallmark characteristics of recent versions of HPAI that have been seen in Alaska. But I do think there's a lot of overlap between these symptoms. So in general, you would never want to just assume what something is from the outside. So agreed that you definitely want the proper PPE and training before you handle anything. And as Rob Kaler would remind us, you also need a sample freezer, dedicated sample freezer that is not where you store your food items before you collect anything, so you can make sure to keep it cold and keep the cold chain, so we can have good samples to analyze later.
Yeah. Another concern for marine mammals would be something like rabies as well.
Mm-hmm.
Having similar symptomology.
Yeah. Thanks, Kathy. Yeah, I feel like that's good as we're all headed out to field work, and we might see a lot of things while we're out in the water this summer that there's a whole variety of things that could be impacting marine mammals and seabirds. And so just to like keep that in mind. And Rob has added, Rob, I'm assuming those are some symptoms specifically for HPAI. Is that right?
Well, I think there is a lot of confusion between harmful algal bloom exposure to biotoxins as well as HPAI. As Sarah said, there's a overlap in the behaviors. So if they're swimming in circles, or they're like kind of pecking at something on the beach that really isn't there, head bobbing, and then lack of predator avoidance. Those are, I think that's all, harmful algal bloom exposure as well as bird flu. There's a lot of overlap in the behaviors.
Also important to note that we often see multiple things in a bird. So just because, in addition to having some of these behaviors overlap, you could also see DA, saxitoxin, and HPAI in a bird. That's not unlikely.
Yeah, and just to layer in on that, they might be exposed to harmful algal bloom biotoxin, and then now they're more susceptible to bird flu or vice versa.
Mm-hmm. Yeah, most MERS in particular are exposed to avian influenza. You would expect something like 50% of wild MERS to have had some kind of AI exposure, but not necessarily high path. So that's not unexpected at all. But yeah, like Rob's saying, the immune system could be compromised, and these stressors are all kind of cumulative. So it's hard to know in advance, and it's hard to know after what killed them.
Yeah, and in a warming ocean, less food, so they're compromised in that way as well. So also more susceptible to either bird flu or biotoxin.
Okay, well, Kathy and Sarah, thank you guys so much for joining and sharing this information.